Becky was on earlier this morning filling in for somebody else. She's the Director of Software Solutions at the University of Michigan's Department of Nephrology. She's been involved in the kidney research community for a while and was one of the lead developers of the Kidney Tissue Atlas Ontology, KTO. Obviously that's an important, relevant research project for the initial tissue of kidney here in HubMap. So Becky Steck.
Great. Can everyone hear me and see slides?
Yes.
Yes.
Oh my gosh, that's awesome. 2 for 2. That never happens. So thanks again for actually for letting me speak to you all today. So this presentation, I'm going to be focused more on the Kidney Tissue Atlas and some of the other pieces of software we are building for the KPMP and kind of how we're going about doing that. So I'm going to buzz through some of these slides quite quickly because many of you saw them already this morning, but just a reminder that one of the big outcomes of the Kidney Precision Medicine Project is this Kidney Tissue Atlas, where we can not only have lots of information, lots of spatial data, but also linking that data to different molecular and clinical data so that ultimately we could come up with new treatments and hopefully new outcomes, if you will. So, and again, another refresher slide, just a reminder that there's a lot of different data that we're dealing with. But in addition to all of the different types of data that we're collecting, it's also quite a complicated system. I think everyone probably is going to show a slide like this, right, to explain all the different data that we have and all the different tools that go along with it. So Oliver earlier today showed the slide as well, but so just to walk through it quickly, we have a lot of different data that is being generated for this project, and then we are trying to put it all in a central place, the data lake, at which point it's kind of validated, organized, curated, wrangled into the knowledge environment. From there, we want the knowledge environment to be the place that actually serves up data to a variety of different in addition to the Atlas, a variety of other tools as well, right? We just heard about with HubMap that, you know, some folks just want access to the data sets. So actually the next slide goes really well into what Katya was just saying as well. We have also a variety of masters that we're trying to serve, right? We would call these different user personas. So we know that one of the types of groups that we need to serve with the KPMP data and the project as a whole is patients. And so, as well as pathologists, clinicians, and a variety of other types of researchers. And those are just our main ones. There's also, you know, students and fellows and pharma, you know, folks that are trying to develop new drugs. Those are all different people that we can conceive of that would benefit from the things that we're trying to build. And I'm going to go back here for a second to just say that, you know, even though ultimately the Kidney Tissue Atlas is one of our big things that we are needing to build, the software development team is in KPMP needs to build a lot of other things too that are pretty much all of these other boxes. So for instance, we need to build the place that holds all of that data in a central repository, and we need to build the pipelines to get the data into that repository, and then to normalize it and curate it and validate it to have it sit in the knowledge environment, which also is something that needs to be built. So there's a lot of software activities here, which is why I wanted to show you a little bit about our process. I'll take you through this. I'll let us sit in the slide for a little bit, but in essence, we say that we have a user-centered and agile software development methodology, and a lot of that is it starts with first understanding who our user is, which is kind of that slide I just flashed up earlier with those different personas. That's one way that we do that, is we interview a variety of different users and try to understand who they are, what their needs are, what they do. And that gets us really to then the next piece, which is defining the problem. So based on, you know, who we understand to be somebody that needs this tool, what do they need it to do for them? You know, what exactly do they need it to do, and what can it not do as well is also very important. From there, then we do we do a lot of prototyping, ideating, a lot of whiteboard sessions, right? These hopefully are not unfamiliar to anyone on this call. But you know, we have oftentimes what happens is we have somebody on our staff who his title is a user experience designer, who will interview these different people that represent those users. So for instance, we recently built a tool that involved transcriptomics data, and we talked to the transcriptomics data people from the KPMP to ask them about what they're, what they need and what they would like for this to work on. And we whiteboarded with them. We took some mock-ups and went back to them for subsequent calls and continued to refine the software. And that's really what that evaluate step is, right? We need to, instead of just talking to people once and then going off into our fortress of solitude, we need to continue to work with them and learn from them. So we will put those prototypes back in front of them. We will, try to put real data in those prototypes, because a lot of times we found that seeing a mock-up or a whiteboard is great, but until you have the real data in front of you, there's a lot of new things that come up at that point. And then when we actually approach building our software out, we try to release often, and we try to release small pieces of code so that nobody has to wait a year for the great unveiling of a big piece of software. We try to carve out useful pieces right away and get those in front of our users as soon as possible. So I'm going to talk a little bit about some of the internal tools now that we have built, if I can get my slides to go. So I showed earlier this morning this concept of the data lake uploader, which was the reason I showed it this morning, was to talk about how it's dynamically presenting different forms based on the required metadata for a different data type. But there's a variety of other tools that we have either built or in progress of building, and we do our best when we build these tools to follow all of that process that I just listed. So we have a tool, actually the first two were mentioned this morning as well, specimen tracking tool and the RedCap instance that's covering 8 different sites that are doing biopsies. We also have set up a single sign-on service because we recognize that there was a variety of tools that all needed login. So we're using Shibboleth and InCommon to support that. We have a user portal being built right now. It's actually it's in use for some of our apps, but needs to be extended for others, which in essence allows us to have different roles. So for instance, in RedCap, different people have, certain sites should only see certain sites worth of data. So we need to institute the idea of roles in all of our internal applications. And then another one that's you'll see some hints of later in my presentation, is our digital pathology repository, which is a way for all of the pathologists to review the whole slide images that are being created from that diagnostic core. And we want them to be able to annotate those images. Here's glomeruli, all the different glomeruli, and then score the features of those slides is a big part of our project. And then there's also some really cool stuff that you'll see a little later, where the digital pathology repository is where we're going to first build out the ability to do image segmentation, which really the way me, a non-image segmenter, would describe it is the ability to identify a feature in an image and then see that feature elsewhere in the image or even in other images by breaking it down into pieces that can be recognized. But if we go to the external tools, there's kind of a couple main camps for our external tools as well. One is the participant-centric tools. So we have the remit of making sure that our patients that are providing biopsy tissue to us, providing a sample from their body, that we can try to support them in the return of results wherever we can. And so one of the ways that we know we want to do this is by allowing them to actually look at their biopsy images. I think some of the pathologists were surprised when our patients said they wanted to see those images, but they just want to kind of understand what their body might look like and what other people are using these images for. So we have built, and I have a link to it in a later slide, but we have built a demonstration version of this web-based slide viewer where we have catered kind of the content to a participant as opposed to a pathologist. So in this case, if you can squint, you could read that down here we just explain that what they're looking at is a trichrome stain, and here's why we do those, because they're helpful for detecting scarring. And so that's an example of some of the things that we're trying to build interfaces for our participants. Other things that we're trying to build for our participants include just some summary stats about what's happening in the study in general, how many people have been biopsied, how much of that data has been analyzed. Another thing that seems to be kind of interesting to our patient panel is, where did my tissue go? So I was biopsied in Cleveland, and then somebody mailed a piece of my tissue to California where there was a proteomics analysis done on it. You know, that kind of information is very interesting as well. And we're in a lot of discussion right now to learn how much additional data we can return to patients. But we expect that somehow we would at least give them certain, a compilation of some of their clinical data that has been collected during those visits. But for the kidney tissue atlas, that's where I want to park for most of my time here with you. One thing it's a helpful frame of reference for everyone on the call to recognize is that we envision that this is an atlas that needs to be something that people can enter, if you will, from multiple places. So some people might go to the kidney tissue atlas because they have a favorite cell type, right? They love the podocyte and they want to search for what we know about the podocyte. Some other people might be more molecular minded and have a favorite gene that's of interest to them, and they might want to search for that. Similarly, you know, a pathologist might want to look for a particular feature from the slides, right? I'm really interested in fibrosis, and I want to see what information you have about that. And lastly, of course, clinical features as well. So one thing that has been helpful for us as we consider what to build and in which order to build it, it was just kind of recognizing these 4 different entry points. So now we've been starting to pick away at demonstration, proof of concept, prototypes of some of these different entry points. And so that's what I'll take you through now. So what we've been doing is accumulating all of these different demos into one place here. Unfortunately, this is only accessible right now internally for the KPMP consortium members, because a lot of the data in here is not validated. But these proof of concepts I'm going to take you through here next. So one is, this is showing, these screenshots are from the mock-ups that I was talking about that the user experience designer would create. This is one of our ideas about what might happen if somebody was interested in a particular cell type. So we're still working out the controlled vocabulary, so the organization of those cell types and what cell types we show, that's not set in stone at all at this point. And the nephron schematic is the same story, but this was a nice proof of concept. So what can happen here, and we've built this tool out, is that you can select a particular cell type. And right now, what we present to you is the transcriptomics data that we have from the 3 different sites that are doing single cell and single nuclear transcriptomics data analysis. So that's one example of something that we envisioned for cell type based searches. And then you can see we have this idea that we could also add to that. Right now we just built out transcriptomics, but you could envision also clicking on a cell type and seeing the proteomics data that we have, for instance. Another type of, the next entry point that I wanted to show you is this molecular based, right? So if you have a gene maybe, so that's what we've built most recently and just unveiled last week at a face-to-face meeting for KPMP. So this is, again, here are the mock-ups, and I'm trying to fill you in with all my caveats. In this case, the caveat is we're still working on the appropriate level of granularity to show of cell types here. But the idea being that if you search for a gene, you could see expression values in that gene across those different transcriptomics data sets as well. And it's really been very powerful, even just as an internal tool so far for this past week, as an internal validation of our cell types that we're annotating our clusters with. And we're really learning a lot just being able to see 3 data sets at once. So you search for a gene and you get 3 violin plots stacked on top of each other all at once right now. But let's move on now to the idea of image-based search concepts. So, and there's that link I talked about earlier. So that's a link actually that you could get to. And so after I talk, I'll put that in the chat as well, unless somebody beats me to it. So you could see that we've built a web-based slide viewer. And I'll also mention that code is on GitHub and it's accessible to all. So what we're doing with this slide viewer right now, it's really just fun to play with, honestly, to go in and see how well the response is to the web-based tool. But what we're really working on next is this idea of using then machine learning, as I mentioned, with that segmentation. So here I'm going to show you a video if it works. This is a proof of concept, but it's an example of one of the things we could envision doing with this is that maybe we could have it be that you hover over particular structures and then it highlights the nephron schematic component that you're on, right? So if it's you can maybe glomerulus, everyone on this call might be able to recognize on a slide, but could you tell your proximal tubule apart? So that's an idea. So again, proof of concept, but something that maybe we could use to map these histology. We could, use common this machine learning to look at different kidney structures on the images and then and recognize where they are. And that video is just looping. So here's another one. Now this is very much a proof of concept as well. Sorry for all the caveats, but this is just, oops, I went too far. I didn't press play. Here we go. This video is showing, so a couple things as this video plays. Please recognize these genes are not based on KPMP data at all. And the structures that are lighting up, that hasn't been validated yet with our pathology team. But here's just an example of, even with all those caveats, maybe a user, you can picture how a user might be able to interact between spatial and molecular data, right? Maybe instead of how before a user hovered over a glomerulus and it lit up the nephron schematic to say that's a glomerulus, maybe instead it lights up the genes that we have identified to be highly expressed in glomerular tissue. And that, that's an example using maybe in a reference, kind of a normal setting, but KPMP, as you remember, is all biopsies from different diseases. And so maybe we can start, some kind of the grand vision is we can start clustering different patients based on their molecular, signatures, and then we could look at those patients' slides and put the two together and see if there's relationships. So there's a lot of really great things that we're thinking about, but I would say it's very helpful for us is to, as you have to eat the elephant one bite at a time, it's been helpful for us to consider these different entry points. So with that, I'm going to stop and just acknowledge that there's a lot of people from KPMP to acknowledge, but since I just gave a lot of software demonstrations, I thought it important to acknowledge all the different people that have been building software, either as part of the internal tools or the external tools. So the software team is all listed here. There's many from the University of Michigan that are working on the Kidney Tissue Atlas, and then the internal tools are both folks from the University of Michigan as well as the University of Washington with the Data Coordinating Center. Okay, thanks. I'll stop talking now and let's have to take some questions.
Thank you, Becky. Are there any questions? I have a question for you, Becky. Your prototype users.
Yeah.
What if the prototype user is a robot?
So
that would be the most important prototype user.
Yeah. So so a couple things I'll say about that, and maybe they're not the right things, but one part is I should call out, because I didn't really talk about this user very much, but we do envision that as part of KPMP, one of the reasons we want to make this data available is for lots of other people to consume it and use it and learn from it as well. And so that is why this particular user here of someone that might develop their own tools. So we do plan on making our data available from an API, which maybe is the robot you're talking about. Yeah, and so that is one of the users that we need to serve. And if I go back a slide here, that's one of the things that we're getting at too with this idea of, in addition to the atlas, there might just be a raw, what we're calling raw data here, but not necessarily raw as in the least processed form, but more so just, just give me access to the data sets from the back end with no user interface component on top of it. Did that answer it?
Yes, thank you.
Yeah.
Yeah, same.
Yeah, go ahead.
Go ahead, Sam.
Hold on, I hear a siren going by. So,
I knew it was you.
So you talked about, having the data be accessible and the tools being accessible. How well could I take the HubMap data and just use the tools that you've already used and developed?
Right. Right now, not probably easily, but it depends on the data you're talking about. So there's a couple ways that we're envisioning this could happen. So one is that we think there's some people who may want to put their own data into our data structure so that it's served up in the tools, right? So let's take an example here that, like, imagine there's a fourth data set down here that you could put violin plots for, right? That is something that I would say is a concept we understand, but it is lower right now on our priority list. However, and maybe I'm not sure if I'm answering this the right way, but there's also this idea that we have that we do want to make sure all of our data and code is shared, right? So for instance, our metadata standards are going to be out there, and so if you were to organize your data to our metadata standards, it would be easier for us to consume that. It's just not so we're not shutting the door to that. It's just not it's not the app we're building right now.
There would need to be some sort of map from Hub Map to KPMP.
Yeah. Mm-hmm. But I think we're trying to make it as easy as possible to make that map.
Right. Plus, if I may just add that it has to go through the rigorous quality control criteria that have been lined up for accepting any data from outside. So,
yeah. And Sanjay's our quality control guru, so that's well put, Sanjay.
Got a question in chat as well.
I've got a chat one. Oh, this is for Simon.
It is, yeah. Oh, yeah.
It's for Simon.
Got it.
Okay. Thank you for looking.